How is coronavirus diagnosed? Coronaviruses are RNA viruses. They use RNA instead of DNA in their genetic code. SARS, MERS, and COVID-19 are all beta coronaviruses. By sequencing the genomes of viruses, we can compare any new sequence with other described closely related strains. Finding the closest match to the new strain will likely provide information about the original host from which the new virus jumped to humans, as well as the geographic origin. DNA is a self-replicating material which is present in nearly all living organisms as the main constituent of chromosomes. It is the carrier of genetic information. RNA, on the other side, is a nucleic acid presented in all living cells. Its principal role is to act as a messenger carrying instructions from DNA for controlling the synthesis of proteins, although in some viruses, RNA rather than DNA carries the genetic information. Genomes are the haploid set of chromosomes in a gamete or microorganism, or in each cell of a multicellular organism. Here you can see a comparison between RNA and DNA. RNA is a nucleic acid presented in all living cells. Its principal role is to act as a messenger carrying instructions from DNA for controlling the synthesis of proteins, although in some viruses RNA rather than DNA carries genetic information. Genomes are the haploid set of chromosomes in a gamete or microorganism, or in each cell of a multicellular organism. Accurate diagnosis of COVID-19 is essential for epidemiological studies. One way to identify COVID-19 is to sequence its DNA and to compare the sequence to other confirmed COVID-19 sequences. However, as an RNA virus, its genetic code is written in RNA. So sequencing an RNA virus requires that it first be converted into an RNA-DNA hybrid that can be sequenced through a process called RT-PCR. And this stands for reverse transcriptase polymerase chain reaction. RT-PCR test kits for new pathogens are tricky to design and to implement. They must be custom developed at the start of an outbreak and must not cross-react with other similar strains. They must also show positive results when the patient is infected with COVID-19 and negative results when they're infected with anything else, as this is challenging. Once developed in the case of a pandemic, these test kits must be mass-produced and distributed as quickly as possible, which is a logistical nightmare. Once a new epidemic virus is identified, the world's health resources work to create a diagnostic test with the national healthcare resources of developed nations. In this case, a group of German researchers developed an accurate test that was adopted by the WHO for worldwide distribution. The CDC in the US decided to develop its own test kit, but unfortunately, the first batch of these tests was unreliable due to some unspecific manufacturing errors. Given the logistical problems with mass production and distribution, testing should not be done on asymptomatic people who have not had any contact with infected people or have not traveled to infected areas. During an epidemic, resources are strained and our healthcare system needs to focus on the people who are sick, not the people who are simply worried. The epidemic in China has already outpaced its ability to produce a sufficient number of RT-PCR test kits and is now using CT scans for diagnosis. Testing in different countries will rely on individual national healthcare systems, but all will use some sort of RT-PCR test for definitive identification. A study just published suggested that CT scans could be more effective than RT-PCR for a diagnosis of COVID-19, especially in areas where the disease is epidemic. However, a chest CT scan cannot definitely prove that an infection is COVID-19 is present. But for the purpose of patient survival, the chest CT scan can guide to an appropriate treatment. In epidemic areas, it is crucial to provide fast and accurate diagnosis so that the patient can be quickly isolated and treated. Sequencing RNA takes time, but possessing the sequence is the gold standard of definite identification. Besides the genetic sequence of a virus are hidden inside the virus external envelope. Our immune system cannot attack what they cannot access. So the RNA sequence of a virus is not the target of our potential vaccines. Let's take a closer look. See how the RNA is protected from attack? So why don't they just make a vaccine? Vaccines can be produced by injecting an inactivated, basically harmless, virus into healthy individuals, who then mount an antibody response much more quickly because the immune system has seen this invader before and is now ready to launch a rapid response. However, the development of vaccines is time-consuming since they must be developed, tested, and mass-produced in a process that will take more than a year if all goes well. We can also use antibodies to detect whether a person has been infected and subsequently recovered. So the RNA sequences can be used to positively identify and classify COVID-19. CT scans can be used to diagnose sick patients and guide treatment in epidemic areas. And antibodies are the source of your immunity and could be used to trace the epidemic. Of these, RT, PCR, and immunological tests rely on the unique sequences of RNA and proteins, respectively, or the COVID-19.